who kindly accepted to talk today. So Richard Cooper, he said, has a background in crystallography and computational chemistry, but he's now head of machine learning and method development at Oxford Drug Design. And he's also associate professor of chemistry at the University of Oxford. And his research interests lie in mathematical modeling and analysis of molecular materials, including optimization of models used in crystal structure analysis, prediction and rationalization of solid-state structure, and intermolecular interactions. And he has a particular interest in the use of machine learning tools in small molecule drug discovery. And you can see the title of his presentation on the slide is showing now. So the floor is yours, Richard. Thanks again for accepting to talk to us today.
Thank you, Nicholas, and thank you very much for the invitation to speak. Thank you, everyone, for coming. The title of this talk is Guardrails versus Guide Rails, or I can't remember, maybe it was the other way around. But I want to talk about useful generative AI models in drug discovery. And so we're going to get there by talking about when generative AI models are less useful. We'll come around to that and ending up at that point in the talk. So in case it's not clear, maybe it's not clear, I wanted to put your minds at rest and explain what I mean by guardrails versus guide rails, very subtle difference in sound. But the idea is sometimes an AI model is producing lots of output and we need guardrails to prevent it kind of crashing into the sides. That's the guardrail analogy. Whereas if we have a model that's running on guide rails, it can only explore, it can only go to places that make sense, chemical sense or physical sense. So yeah, that's the analogy, and you'll see how that fits in as we get really to the third part of this talk. So a little bit of context about where I work. So this is Oxford Drug Design Limited. It specializes in computational drug discovery tools, and it has in-house projects targeting tRNA synthetase. We're based in the middle of Oxford, right opposite the Bear pub, if you know that. So that nice building in the middle is where our offices are. And this is the team, just to say thank you to them now rather than at the end, in case we've run out of time or I forget. So there's the computational team, Anthony Chubb, Paul Finn, me, and Aras Assad, who have kind of developed most of the things we'll see in the talk today. There's a couple of other people that I'll check as we go through. Right, the next slide, I feel almost embarrassed presenting this to this audience, but just kind of to set some context. We know drug discovery is a hard problem, and we know there's some kind of pathway like this, but we also know it's not really a linear pathway. You keep going back and starting again and going all over the place. But what I wanted to distinguish really is that I feel like this BioSim community is probably more dealing with pathways and target ID and also docking, I suppose, a bit to some extent, whereas what we're interested in is usually hit ID, lead optimization. So there's a slight difference in where this talk is coming from. So I thought it was useful just to say that at the beginning, so you're not kind of thinking, when's he going to start talking about biosimulations and that sort of thing. So we're very much focused on hit ID, lead optimization, and that kind of thing. Right, then the other elephant in the room is AI itself. So AI has this immense potential, and it's also currently in a phase of massive hype. And the problem with that is it's very easy to overpromise results to customers, to investors, and increasingly, even in the last few weeks, we're seeing governments starting to believe in magic efficiency savings that AI can do. There's an example from the recent news. There's some news stories there about this company, Builder.ai. And this is less of a, well, I suppose it is an ethical problem, but it's more of a fraud problem, where the AI model didn't really do what it was saying, or that they slowly admitted this as they went along. This platform was supposed to write code for you, and it turns out they were just employing hundreds of real programmers to pretend to be AI models and chat to users and write code for them, just so that they could kind of build up their value and hype it up to investors. And they got found out, and that there are all the news stories. You can judge for yourself what happened. But that's the danger of hype. In terms of kind of back to the kind of basics level, what we actually do when we're trying to solve problems. So away from the ethical problems and the investor problems, we've still got to make progress. We still have to do something useful with AI. And we can end up in a scenario where there is a kind of a knowledge gap represented here between the kind of experts in statistical methods and the experts in, say, biology or chemistry. And on either side, lack of knowledge of the domain on the other side can lead to people making mistakes. So what I'm going to do, well, so I guess the thesis of this talk is that we can only make progress by bridging that gap and avoiding the hype. So I'm going to talk about how we assess new methods and give some examples of kind of when it goes wrong and maybe how we can learn from that and avoid from it. How data sets can be biased and where we can learn from that. And then finally onto the generative AI bit. I've said, do we have sufficient information? That's maybe the first example. And then I'll talk about the guardrails versus guide rails approach and review some bits of literature and tell you what we're doing at Oxford Drug Design to have a slightly different approach to building generative models. Okay, so those are the three parts. So, oh yeah, this slide is in for people that don't know about molecular representations. So obviously, I guess everyone here does. I don't need to say we represent molecules. To cross this gap, we basically represent our chemical and biological knowledge. It ends up being represented as a vector of numbers, some kind of representation of the physical system. It might be fingerprints, it might be things to do with the shape or the molecular graph or something like that. But in general, they all end up as vectors of numbers that go into the mathematical side of the problem. We know that. Okay, so section 1, assessing the effectiveness of new methods. So I want to talk about this paper, Posebusters, which came out last year, and how it kind of took a fresh look at some AI-based docking and showed that the methods are maybe not quite as useful as they announce themselves to be. Then another example where there's just some results that look too good to be true and how we've investigated that. And another example where results look too good to be true. So these are, how do we go about assessing these methods? What things do we need to do that? So Posebusters, so I should say this is nothing to do with me, but it is from some colleagues at Oxford, published by Buttenschern, Morris and Dean in Chemical Science last year. And they assessed 5 different deep learning docking methods that all had it's kind of similar but kind of different deep learning technologies underneath them. And there's on the slide there's a description of how they each tackle the problem. And a typical claim in a paper is that state-of-the-art performance in terms of RMST of atom positions in a docked molecule to experimentally measure positions in a crystal structure. So that's the measure they're using to claim state-of-the-art performance. And when you dig into the results, you find that, or that they found, Martin and Garrett and Charlotte found that despite these claims of state-of-the-art performance, there's still a lot of kind of nonsensical stuff coming out in the docking poses. So they often produce physically implausible molecular structure. And there's a lot of these in the paper. You can see I've picked examples F and H and stuck them on a slide here from the paper. So there's plenty more before that. But here we have like a double bond that's not flat. So there's a problem with the molecular geometry, and that can't possibly be the real dot pose because it would have too much strain energy. The second example is a clash with a protein. So there's I think it's a methoxy group poking into the protein in through the kind of surface. So there'd be a really high energy penalty for that, but the the method is just kind of ignoring that and telling you that's the docked pose. So what they took away from this is that although the RMSD metric is useful, the physical plausibility of the models and the ability to generalize to other examples, didn't really give any examples of that, are no better than classical docking tools. So when you take these extra things into consideration, these AI-based methods don't perform any better than classical. It's quite a important result, and it's important not to knock down AI, but because I think if we want to make progress, then we need to be honest with ourselves about what these methods are doing, and try and actually improve them and make them do what we want them to do. And I'll come back to that in another section of this talk. So that we have to evaluate things on the right metrics, and of course that requires domain knowledge of the chemistry and biology in this case. Oops. Yeah, I've said that. Good. Gone the wrong way. Okay. Second example. This was uncovered by a doctoral student who's working with us at Oxford Drug Design. He's a mathematical student, and this got flagged up because someone was reviewing a paper that we wrote, and we'd claimed a certain accuracy, and they'd said, "Oh, well, haven't you seen this latest paper? It's got an accuracy, a rock walk area under the curve for a classification problem of bigger than 99%, 0.99." And so that sounded a little bit suspicious to us, because the previously best published example was this example TrimNet, which had this area under the curve of 0.851. If you're not familiar with area under the curve, it's a metric where you rank your molecules, and you've got plot your true positive rate against your false positive rate. And in an ideal world, you go straight up one axis and along the other axis, and you get an area under the curve of 1. In a completely random world, you go up the diagonal and have an area under the curve of a half. So you can see that 0.99 would be a quite good, but also quite surprising result. So Alex dug into the paper. The good thing was that the code and data were available, so we could just rerun the model. Alex determined that the metric was not appropriate for a binary classification problem. So there's a fundamental problem with understanding the kind of mathematical statistical side of what's going on. And in addition, there's a fundamental misunderstanding or lack of surprise that the accuracy has jumped from 0.85 to 0.99 in one go. So Alex said that actually it doesn't really matter with this case. Using this metric, the model doesn't need any skill at predicting at all, because of the metric, the way they've used the metric. You would always get 99% accuracy. So we recalculated it because they provided the code, and it's nowhere near as impressive. It's much closer to this random line of 0.5 to 0.6. Now so that's kind of annoying, a little bit of a slows you down. And my argument is that's really a drag on the field, because people are stopping publications because they're saying, oh, well, someone's already done this, it's got 99% accuracy, and you have to then go re-implement the method, prove it's wrong, and resubmit your article. So it's so these things are not just kind of annoying weird things in the literature. They do have an effect on the rest of the field, because it is very difficult to reproduce these methods, or it requires a bit of effort, I should say. And the final one is a kind of silly example, but in the formulation step, this is a published model for determining whether two crystals will co-crystallize together. So you might do this with a drug molecule to try and make it more soluble or more stable, or there are various reasons for wanting to tune its properties by mixing it with other molecules. And they classified the likelihood of it forming a co-crystal based on some experimental data set they had. They used a novel representation, which was just to take a picture like that one there on the screen and train a CNN to classify whether that's going to form a co-crystal or not. And the paper reports a ROC-ORC of 0.925. Now, I haven't said what the paper is because we're still in discussions with the authors. We're going to fix this problem. And here's the problem. So they had to use data augmentation because they had a class imbalance. And the way they did it was to rotate copies of these images of molecules to augment the positive class, the ones that formed co-crystals, because there were many fewer examples of those. And this image, like the one on the right, is typical. When they rotated it, they left these big black backgrounds where they'd rotated the image. And obviously, it's quite easy for a CNN to detect those black backgrounds, and then it knows that's a positive result. There was another problem with data leakage that we always found. That we found in all the examples as well. So these are problems with kind of performing the machine learning in the right way. But again, it makes something look better than it is, and means that everyone else has to play catch up and explain why that wasn't quite as good as, why it isn't better than our result, even though it appears to be. So we've seen postbusters using a kind of slightly incorrect metric that doesn't kind of address the proper problem in the domain where it's set, and the interesting result then that those correct results don't outperform classical docking. The deep learning results show the importance of publishing code, which meant we could re-implement, but also of using the right metrics and knowing what you're doing, and how that, it's just I'm annoyed because it was us that it hindered, but this is how you discover these mistakes. But these things do slow down research. And then finally, this last example, just some data leakage and unintentional biasing of the data. Okay, then moving on to some other examples of bias in the second bit of this talk, we've got two examples of data sets that are commonly used that can suffer or do suffer from bias. The first is the directory of useful decoys extended, which you've probably heard of. It's a very common classification data set used to test docking programs, but it is frequently misused. And then CASF 2016, which is, I can remember what it was, something. No, I can't remember what that stands for. I think it's on the next slide. But this is a training bias problem that crops up from time to time. So DUD as a dataset was designed to help benchmark molecular docking programs by providing challenging decoys. So it has 22,000 active molecules and their affinities against 102 targets, which gives it an average of 224 ligands per target. So you can see why this is such an attractive data set for people doing AI, because you've got tons of data to train your model on. And then on top of that, for every active, you've got 50 decoy molecules, which the DUD authors propose probably don't have activity. They don't know that for sure, but they've given them a different 2D topology, but matched the physicochemical properties. And so there's some kind of wiggle room there. But again, it's a lot of molecules. And it's this last step, this generation of the decoys, that actually causes the problems for AI-based or machine learning-based methods. Because if you take any method that isn't doing docking, basically, and train it to classify these DUD results, they will give amazing results. So here, this is from a paper. I won't say again what it's from. Using a clustered cross-validation on DUD, we achieve an average AUC ROC, area under the curve of the receiver operating characteristic of 0.92, and a 0.5 ROC enrichment factor of 79. So that means if you look at the very early enrichment of when you rank your ligands, that you're going to have 79 times more actives in that little section of data than you would expect if they were random, which would give you an enrichment factor of 1. So this method's doing really well at getting all the actives at the top of the list. However, the problem is that Duddy, when it was generated, used molecular fingerprints to select these decoy molecules so that they have similar physical chemical properties, but they're unlikely to bind as well to their given targets. And because of this, any classification method that uses a molecular fingerprint can very easily distinguish between the two. And so that's going to probably include any AI method or machine learning method that you can think up, apart from docking. So not only this, the Duddy website says it's designed for 3D molecular screening methods, not for 2D methods, such as molecular similarity. So they say it straight on their webpage. And there's a paper here that I'll refer you to that I found some of those examples and quotes from. Hidden bias in the Duddy dataset leads to misleading performance of deep learning in structure-based virtual screening. That's from 2019. So it's primarily looking at CNNs because they were popular at the time. But the reason for the bias applies to any model that captures atomic environment. So any type of similarity measurement, any fingerprint method. And so, yeah, so there's the link to go and check out that Duddy. Right, CASF stands for the Comparative Assessment of Scoring Functions. That's what I couldn't remember earlier. So this was a challenge sort kind of to do the community to come up with good scoring functions for molecules docked in a protein target. And you just have to predict the affinity given the 3D structure. So CASF 2016 defines 285 protein ligand complexes as a test set, and those are the ones you have to predict. And if you do well on that, then your method does well in the CASF challenge, if you like. However, what they don't do is provide a training set. So some of these available data sets, they'll give you all the data and you can use that. But in this case, you have to go and find your own training data. So what a lot of people do is go to the rest of the PDB refine set. They take out the test set, those 285 or 286 complexes, and they just use the rest as training data. But when they do that, so they're left with 4,638 complexes. A very small number of those, 102, 2.1%, have identical small molecule inhibitors. So they probably shouldn't be in there because you kind of have an easy prediction of affinity if you already know how well something binds to another protein. 994 of them, that's a fifth of the data set, have identical protein sequence. That means it's the same protein, just with a different molecule bound in. And again, once you know the kind of range of where your binding affinity is going to be, then just predicting the average is going to give you quite a good result in any statistical challenge like this, because you've got somewhere to start from. Then 32% of the complexes, which is a superset of the 994, have greater than 90% sequence similarity. So they're probably again going to have very similar binding affinities for a given molecule, or binding affinities in general, a range of binding affinities for the molecules that they see. So that means all of these methods using the rest of the PDB or PDB bind to learn or to predict the test set are going to do quite well. So to test whether this, what kind of effect this has on the data set, we got Marco Albanese, who's a computational chemist at Oxford Drug Design, to duplicate the training data. So any complexes with highly similar sequences were removed from the test set. So we kept the test set. Any training set data that were very similar to the test set were removed. Any with identical ligands were removed. That was just that small number of things with identical ligands. And we were left with just 2,500 training complexes, so down from about 4,500 We call this the debiased training set. And then we came up with a method. It doesn't really matter what the method is or how well it performs. That we used a re-implementation of OnionNet, which was published a few years ago. But what matters is how the scores change as you go from just the refined set with the test set removed to the refined set with anything at all similar to the test set removed. And you can see that the mean squared error jumps up, the Pearson's R correlation falls down. So I've called this training data more challenging, but of course, in the context of this talk, more challenging equals more opportunity for genuinely measuring progress, because I would say it's more important to be able to use a data set to make progress than it is just to be able to publish a result and say, look at our amazing method in the long run, obviously. So those are the two data sets. We've got to be aware of bias in them. The struggle is that debiasing things gives you a more challenging data set, and that makes your headline results worse. So in a short attention span world, it's difficult to kind of get people interested or excited about this and making and publishing this stuff, but you can't make progress without a kind of fair testing model. Right, the last bit then. So the message here, which could be kind of engraved on something, the message is be skeptical of the hype. We've already kind of said that a little bit. Be skeptical of learning things that can't possibly be learned, and be skeptical of designing things that can't be made. And we'll come across each of these in this last section of the talk. So I want to talk about a headline: AI discovers novel antibiotic, and the follow-up publication to that, which kind of starts to question how well it did that. Then talk about what generative AI is doing for drug discovery, and why does it need such comprehensive guardrails to make it spit out useful results. And then think a little bit and tell you a little bit about what we're doing to build more efficient models using guide rails to enforce reality. So there's the words from the headline of the talk, and this bit should sort of bring them together. So here is a paper from Cell in 2020, and the message is, so this is a deep learning approach to antibiotic discovery. The message is AI discovers a new antibiotic, and in a way you can't argue with that, but we'll see why it may have been more luck than judgment that AI discovered this antibiotic. So they trained a message passing neural network to predict antibacterial activity of FDA-approved drugs and a natural product library. So that's the input data. Then they predicted the activity of compounds in the drug repurposing hub database. From that, they found 51 actives, classified 51 molecules as active, and one compound, halicin above, was selected for a detailed study. So that's halicin at the top right, with those sort of 3 sulfurs in a row and those nitrogens and nitros and amine groups. And what did they find? It is a good antibiotic. It has a novel mode of action, which we'll come back to. It doesn't have related chemotypes in the data set. We'll come back to that. And the core reason that we might say, is this just AI getting lucky, is that the sulfur atom is present in 31% of the actives in the training data and 14% of the inactives. So you might start to kind of think, well, if more of my actives have sulfur in, then a molecule like this, which has got 3 sulfurs in, is going to look quite attractive as an antibiotic. But I think we know that just putting sulfur in molecules doesn't make them into antibiotics. So on its own, that's just a kind of chance finding that, it just happens that this molecule's got sulfur in and it is an antibiotic. Now, of course, there are a lot of sulfurs in the actives because there are a lot of beta-lactams amongst the antibiotic molecules, and they've all got sulfur in. And if we take the beta-lactams out of the training data, then the situation is reversed. So sulfur is now only present in 10% of the actives, and is present in 13% of the inactives. And surprise, surprise, halicin is no longer predicted to be active. So we can argue this is an artifactual result, and that this is just being classified as active because it's got sulfur in. And that ties in with our other observations that it's got a novel mode of action, because we don't teach the model anything about the mode of action. So why would a molecule it picked have a different mode of action from the existing stuff in the database? And why would a new chemotype be active in this case? So yeah, so it looks like it's artifactual, and nothing in the dataset really supports the activity of this proposed structure. So it's a case of a study getting lucky. You happen to have found a molecule that's active amongst a set of molecules, and it happens to be predicted as active just because of its composition. There's a paper that breaks that all down in a more detailed and elegant way than I just have. Then when it comes to AI, results are often presented as disruptive or revolutionary. Here's a quote from Variational AI that said 100 AI-generated molecules are worth 1 million molecules in a high-throughput screen. Now, that's not really a testable hypothesis, so we'll just put that to one side, but maybe think about it in the long run. And deep learning enables rapid identification of potent DDR1 kinase inhibitors. So we can dig into this paper a bit more from in silico. So generative AI works by creating novel molecules sampled from the chemical space of its training data. So the idea is it can just find new molecules that are in that chemical space, sample from that distribution, and maybe they'll have the same properties. So in this paper for GenTRL, which is the one I just referenced, it builds a deep generative model using generative tensorial reinforcement learning. And in the paper, it says GenTRL optimizes synthetic feasibility, novelty, and biological activity. So we'll remember those three things. I've put them in bold. And in the end, they found 4 compounds that were active in biochemical assays and 2 that were validated in cell-based assays. And now we'll see how they got there. So their deep learning model generated 30,000 compounds. They then applied some property filters. Off the top of my head, I don't know what they were, but I don't know, maybe, no, I don't know what they were at all. But that cut it down to 12,000. Then the med-chem filters, that will be Lipinski type things, log Ps and so on, cut it down to just over 8,000. Then a diversity filter, I guess that's to make sure that you don't have lots of the same molecule going through to the next stage. Then a commercial analog filter, cut about another 1,000 out of the data set. And that's interesting because in the paper it says GenTRL optimizes for novelty, and yet we still have 1,000 molecules here out of the original 30,000 that were filtered by a commercial analog filter. So obviously there's something it's not quite being as novel as, or maybe the commercial analog filter is stricter, if we're being kind. Then there's a kinase SOM. I guess that's a self-organizing map filter. I guess that's finding molecules that are likely to target a kinase, and again, a pharmacophore virtual screening. So these 2 steps are bringing us down by another factor of 5, more or less. And we wonder why that's necessary, because in the paper it says GenTRL optimizes for biological activity. So if not for biological activity against the target we're interested in, then what? And then there's some other salmon mapping stage. And then finally, we go down from 40 to 6 in terms of synthetic accessibility. And remember that GenTRL is supposed to optimize for synthetic feasibility. So again, it's surprising that at least those 40 were not synthetically accessible. But they've ended up with 6. Those are the results with the biochemical assays and the cell-based assays. Here are the molecules. Number 1 has got activity against DDR1 and less activity against DDR2, so it's a selective inhibitor. That's quite nice. 2, some selectivity as well. So I don't know. So it's found a molecule. However, the similarity of molecule 1 with ponatinib, which is a commercial analog, which wasn't filtered out either by their model or by the commercial analog filter, has been noted. So I think you can see, there's my little laser pointer. Ooh, here we are. So we can see this bit of the molecule is the same. This kind of alkane, what's that called? One of those is the same. This ring is the same. This carbonyl or amide oxygen here has been made into a ring in the new compound. That bit's the same. This bit's the same, and then there's this kind of solubilizing group in ponatinib on the end. So there's some slight differences, but pretty much in the same series at least. Okay, so that's a kind of deep look at one example. But it happens again and again, this idea of generating lots and lots of molecules with kind of any properties and then filtering them down to stuff you can make, stuff that passes through a virtual screen and gives you the properties that you would expect for a drug molecule. And so I did this a couple of years ago. The last ones are 2023, but we could probably go and find some more. And in each case, they're producing tons and tons of molecules. Here, 500,000 molecules. Here, nearly a million molecules, and only ever really making and testing single-digit numbers of those. And so this is the guardrails thing. So my kind of question is, what's the value of the generative AI in this scenario when you could just feed it a list of 1 million commercially available or easily synthesizable molecules and apply exactly the same filters, and I'm willing to bet end up with still some quite useful molecules. And remember that you might say, oh, well, these are generative AI, we might never have thought of them before. But of course, if you can't make them, then they get filtered out by the accessibility filters anyway. So if we just start from the kind of physical reality of what we can make, then we end up with we're going to end up with a much more efficient process, which I'll come on to in this final section. So so the kind of thesis of this is we're wasting a lot of time, a lot of GPUs making molecules that we never end up really looking at, or we have to refilter them. So we want to use physical constraints to enforce reality. So this is an internal project at Oxford Drug Design, and it works in these 3 steps. We generate multiple synthesis routes to a target molecule. So here's one such synthesis route. We might generate 10 or 20 of these. And we use, at the moment, the AstraZeneca AI ZinthFinder code to do that. But you can use any chopping up code that you want, any retrosynthetic analysis. And then once you've done that, you've got these kind of input nodes, if you like, the reactants that you would use to make the products. And you find lists of reactants that are commercially available that you could substitute in at each of these positions. And then the final step is to search that space of reactants while optimizing a property of the product. And the method that we use for doing that is Bayesian optimization. The model is a Gaussian process model. I'll show a little picture of that in a second. Here it is. So we imagine this is our 3 molecules, and we somehow put them in their own little chemical space. So in our case here, the chemical space is formed by the basis vectors of a derived from an in-house shape descriptor. The important property of that space is that reactant molecules with similar shape and charge distributions are close together, which means that if you move not very far, then you'll end up with a molecule that's, as a reactant, quite similar properties. Then we use Bayesian optimization to exploit that similarity. So it might propose little changes to the where we are in reactant space and pick a nearby reactant in order to optimize a property, or it might explore that active space and say, well, I don't know anything about the reactants over here. Let's go and have a look, try and make something and see what the kind of computed properties of that product are. And that process is all, it's kind of all built into a Bayesian optimization platform. The Gaussian process itself is retrained at each step. And so in addition to these molecules having similarity by being close to each other, the Gaussian process can learn about correlations between each of the dimensions in each of the, I suppose there's 9 dimensions here in this example. So it learns not just about what makes a good reactant at position 1, but what makes a good reactant at position 1 when you pick a certain reaction at position 2. So they're sort of correlated. You can imagine maybe molecular weight of one goes up, the molecular weight of another one has to come down in order to compensate. So there will be these correlations, and the GP underlying the Bayesian optimization is going to capture that and help you efficiently explore that space. And then in capital letters, my last point is that all of these products can be synthesized. Now that should have a little star by it, because of course, like my background's in chemistry, and I know not all things can be synthesized just because they happen to have the same groups on them. So there's a little bit of tuning of reactions to be done, but we're in a much better place than letting Gen AI design a molecule where we've got no idea where to start making it from. Okay, and does it work? It's difficult to tell you that I've kind of invented a blockbuster drug with it, but what I can say is that it does rapidly find good molecules. So this was a little study using HIV GP120 inhibitor, agonist-antagonist molecule, and taking that molecule and trying to find similar or molecules that will fit better than that. So we did just rapid docking. We didn't use any other criteria, such as properties. We used the ligand efficiency of that docking. And here on the right-hand side are a list of the some kind of molecules it came up with. But it gets there very fast. So that was 50 cycles, maybe 100 cycles of docking. And it quickly finds molecules that dock quite well. So obviously there's a reality step to actually do some experiments, but that can all be built into this process with a bit more money and time. Yeah, I was going to say something about these. Oh yeah, no. So yeah, these molecules. What's quite nice about some of these molecules is that they have features in them. So they're not themselves kind of commercial or tested molecules, but they've got bits in that are. So this group here appears in some commercial HIV-1 inhibitors of GP120, and I think this group here does as well. So it's quite pleasing to see. It's not definitive proof of anything apart from these molecules exist and dock well, but we found them quickly in a very small number of dockings, guided by the Bayesian optimization. Right. So in summary then, which I've called the unlearnable and the unsynthesizable, Halassin shows that it's still possible to stumble upon good models without robust predictive models. And so this first drug discovered by AI is really an anecdote versus data problem. It found a good molecule, but we would argue, it's argued in that paper that I showed you, that was probably not deliberate. Generative methods are technically impressive, but they may have a synthesizability blind spot. And when I say may, I say that in a very English way. I mean, they definitely do have a synthesizability blind spot. They're predicting stuff. They say the things are going to be synthesizable, but they end up hardly synthesizing anything and throwing loads of stuff out because turns out to be not synthesizable. So in terms of fair comparisons, obviously difficult to insist on this after the fact, but how can you compare 6 molecules filtered from a Gen AI output to 6 molecules filtered from a library of millions of commercially available molecules, and how would that change, how would that baseline affect the claims of the generative AI? And our proposed solution, it's still a generative AI, so we're still sampling from a space that's similar to our space of existing molecules, but we're doing it in such a way as we're kind of putting the product together each time, and that search is based around the physical task that we have to do, which is to synthesize a molecule. And by doing that, we can guarantee that we can probably make the output that we want. Right, here it was a quick summary. Let's not believe the hype. I think these are more like arguments to, when we're thinking about publication of these methods, we need to ensure, or get journals to ensure, that there are domain specialists involved. So if something's in an AI journal, it needs the chemists and biologists to have a look at it to balance out the peer review, and vice versa. If you're reviewing these papers, you've got to insist on results against more challenging data sets, as annoying as you think you might be for doing that. Be suspicious if the code's not there, or the data's not there, or cross-validation has not been done. Check your data for unexpected biases, and that's very difficult to define what unexpected is, but it sort of comes with experience, and I've showed you some examples of unexpected things that can happen. And always compare to baseline models. So if something is revolutionary, how does it perform compared to what already exists in the literature? We saw that Posebusters paper as a classic example of that. And then finally, ask yourself why a revolutionary method needs guardrails. Is it just churning out molecules for us, and then we're left with the problem of kind of virtual screening all those molecules back to a small number? And this is, I think, key, is to use existing processes as guide rails for the method development. So build the method around what already happens in the industry or in the lab, and then it's going to fit much more nicely. So we end up with results that we can translate to the lab, rather than results we do. This molecule looks nice, but no one knows how to make it. And I will finish there, but I just wanted to say we're always very happy to talk about these methods and collaborate with people. We've got that debiased data set that I talked about. We've got in-house confidential data set, so that's more of a kind of collaboration thing with agreements in place, and the same for our in-house computational tools and catalogs of molecules. So there's my email address. But yeah, we're welcome to, very happy to hear from anyone. Thank you for your attention.
Thanks a lot,